Pharmacodynamics cover all the aspects regarding what drug does to the body. If we break the word pharmacodynamics into pharma and dynamics, pharma means drug and the dynamic means power, which means drug power. The type of drug action are like stimulation, depression, irritation, replacement, cytotoxic. When any drug is the activity of system or organ, we call it as a stimulatory action. Example, adrenaline increase heart rate. When the drug decrease action of any organ or the system, we call it as a depressive action of the drug. Examples are alcohol and general anesthesia. Certain agent applied over the skin cause irritation and interfere with the pain pathway, and due to continuous stimulation, signal conduction decreases and reduce pain. Example, eucalyptus oil. It's an irritable action of drug. When there is deficiency of endogenous substances, they are replaced by drugs. Example, insulin in diabetic patient. It's a replaceable action of drug. Certain drugs like actively toxic to the infectious agent and cancerous cells. Example, antibiotics and anti-cancer drugs called cytotoxic action of drug. There are two types of mechanism, which are receptor and non-receptor mediated action of drug. So the non-receptor mediated action of drug include physical action, which contain osmosis, adsorption, demulants and radioactivity. When 20% mannitol is given to a patient with cerebral edema and acute conjunctive glaucoma, it decreases water concentration by action over osmosis. And charcoal adsorbs toxins and it is used in the drug poisoning. Cough syrups produce smoothing effect in pharyngitis, and radioactive isotopes like iodine 131 emit rays and destroy tissues used in hyperthyroidism. In peptic ulcer, antacids are used to neutralize gastric acid, and the metals like iron, copper, mercury are eliminated by using chelating agents. These agents form water-soluble complexes and excrete out. These are some chelating agents like dimacropol used in arsenic poisoning, ferroxamine used in iron poisoning, and D-penicillamine used in copper poisoning. These are the chemical actions of drugs. Some drugs by inhibiting enzymes in hypertension, captopril and enalapril used, which inhibit angiotensin converting enzyme. There is one drug called allopurinol, inhibits xanthine oxidase required for xanthine metabolism and prevent the formation of uric acid. Some drugs by binding to the transporters show the action. Selective serotonin reuptake inhibitors inhibit reuptake of 5-HT transporters and cause antidepressant effect. Receptor mediated action of drug include agonist, antagonist, partial agonist, inverse agonist. Agonist shows maximum intrinsic activity. Partial agonist show intrinsic activity below agonist. Inverse agonist show completely opposite action. Antagonist show do not show any action. It interferes with the other drug action. Types of receptors are inotropic receptors, metabotropic receptors, enzymatic and nuclear receptors. Inotropic receptors are present in ligand gated ion channels, and these receptors are the fastest one, and the nuclear receptors regulate gene expression, and these receptors took hours to respond, and these are the slowest receptors. Ligand gated ion channels are present on the membrane, and these channels required milliseconds to respond. When inotropic receptors got activated, they open channels. This type of receptors are present in the neuromuscular junction, that is nicotine acetylcholine receptors, and also in CNS, GABA and glutamate receptors. Here in this diagram, when agonist bind to the binding site on the closed ion channel open, and ions like sodium, potassium, calcium and chloride start to flow, and due to this there is depolarization or hyperpolarization and cause tissue response. G-protein coupled receptors are also called metabotropic receptors, and these are present on the membrane. When these receptors got activated, they act on ion channels or enzymes. When agonists act on G-protein coupled receptors, G-protein act on enzymes like adenylyl cyclase, guanylyl cyclase, phospholipase C, and generate second messenger like cyclic AMP, cyclic GMP, IP3DAG. When agonists bind to metabotropic receptors or the G-protein coupled receptors, GDP bind to the alpha subunit, exchange with GTP, and then stable subunit dissociates into alpha GTP and beta and gamma subunits. Then beta GTP bind to G protein. There are 4 G proteins: GS, GI, GQ, GO. GS stimulates adenylyl cyclase and cause increase in cyclic AMP. This cause increase in the heart rate, and this type of mechanism seen in beta adrenergic receptors. GI inhibit adenylyl cyclase and decrease cyclic AMP. This cause relaxation in smooth muscles in blood vessels, and smooth muscles contain alpha 2 adrenergic receptors. GQ protein stimulate phospholipase C, cause increase in IP3-DAG muscarinic receptors show this type of mechanism, and GO stimulate calcium channel. Beta and gamma subunits will act on the enzymes and ion channels, and beta and gamma subunits are also required for the regeneration of stable subunit. GTPase enzyme act on the alpha GTP and replace GTP to GDP and integrate alpha, beta and gamma subunits to form stable subunit. Transmembrane enzyme linked receptors are also called enzymatic receptors. It involves kinase enzyme activity depending upon whether intrinsic activity present or not. There are two types: intrinsic tyrosine kinase activity, no intrinsic activity. So the first one, enzymatic receptors with intrinsic kinase activity. In this case, drug bind outside and tyrosine kinase enzyme activate intracellular. This type of enzymatic receptors are seen in insulin receptors. And the second one, enzymatic receptors with no intrinsic activity. When agonists bind to the receptors, there is increase in the affinity for cytosolic kinase, that is Janus kinase, called JAK. Due to phosphorylation, JAK activate and recruit STAT, signal transducer and activator of transcription, cause transcription and tissue response. Another type of receptor is nuclear receptor, present intracellularly, and these are the slowest receptor. Examples of these receptors are sex hormones, vitamin A, thyroxines, and glucocorticoids. Dose response relationship is the relationship between concentration of drug present at the site of action for pharmacological effect. There are two types of dose response curve, graded and contour dose response curve. When the curve plotted between dose and response and it takes the form of rectangular hyperbola or sigmoid shape, called graded response. The pharmacological effect is said to be present or absent, all or none. This curve is called contour dose response curve. We can appreciate the graded or contour dose response curve, and the example of contour dose response curve is ovulation. Therapeutic index is the ratio of median lethal dose per median effective dose. Median lethal dose is for animals and toxicity dose for humans. Median effective dose is nothing but the dose at which half of the population show effect, and median lethal dose is the dose at which 50% of population died. Basically, therapeutic index is the range or the distance between minimum therapeutic effect and maximum adverse effect on dose response curve. Drug with more therapeutic index gives safety margin, because more the therapeutic index there is wide range of concentration, and when we increase dose it do not cause adverse effect. Drug with more than one therapeutic index is safe. When the same drug is used for different conditions, then therapeutic index changes. Drug with high therapeutic index are penicillin and atenolol, and with low therapeutic index is warfarin, and when we increase dose of warfarin it was adverse effect very fast because of narrow therapeutic index. Drug potency is the amount of drug required to produce effect. Minimum dose required, more is the potency. Drug efficacy is the maximum effect produced by the drug. Therapeutic window is nothing but the range of concentration at which drug gives desired response with minimum adverse effect. Combined effect of drug. Adductive effect. When two drugs are given and the combined effect is equal to the sum of individual drug, called adductive effect. Example, ibuprofen and paracetamol. Potentiation. Enhancement of one drug by the other drug, called potentiation. Carbidopa enhances the effect of levodopa by preventing metabolism of levodopa. When two or more drugs are given and combined effect is more than individual one, called synergism, and when one drug prevents the action of other drug, called antagonism. There are two types of antagonism, competitive and non-competitive. In competitive antagonism, agonist and antagonist act on same side and cause right shift of dose response curve, but in non-competitive antagonism, agonist and antagonist act on different sides, and due to this there is decrease in the response of dose response curve. Thank you for watching. Please like, share and subscribe.